Okay, right, so I had this idea of doing a bunch of medical SBAs as a way for people to learn, really. So I've compiled together about 10,000 single best answer questions, and we're gonna make this series where we just kind of go through them, really. So if you're, if you're a medical student, if you're a nursing student, if you're someone who is, related to that, or if you like, you're just genuinely curious, if you're bored and you just put this on the background noise. I'm not sure why you wouldn't listen to something about movies or something more interesting, a bit more of an interesting podcast, but no judgment here. If you want to learn more, that's fantastic. So I'm quite a visual learner. So as we go through these questions, I'm going to be bringing in a bunch of diagrams to hopefully help explain it a little bit more. And hopefully you can learn something from this. If there's a question that you're, still not understanding, do leave a comment. I will do my best to get back to them. I can't promise I will. I'm quite busy, but I will very much endeavour to do that. But anyway, without further ado, let's just get started. We'll probably start with 10 questions for this first video, and then, I don't know, we'll see how it goes from there. This is a bit new to me, recording stuff and going along at the same time. I usually record something first and then go, doesn't matter, let's get started. Question 1. A 50-year-old patient with epilepsy undergoes routine blood tests. Blood tests show evidence of hyponatremia. This may be related to one of her anti-epileptic medications. Which of the following best describes the mechanism of action of the drug that is causing her abnormal blood results? So is it unknown mechanism, blocks low voltage activated calcium channels, enhances GABA-mediated synaptic inhibition, blocks sodium channels in a use-dependent fashion, or blocks GABA-mediated synaptic inhibition? Okay, so this patient's taking anti-epileptic medication. There's a few that we can start to think of here. You have valproate, you have gabapentin, ethosuximide, and carbamazepine. And those are probably the big four that you're going to want to remember, or at least that's the ones that I'm being told to remember. Good news is none of these work on blocking GABA-mediated synaptic inhibition, so we can rule that out. And thankfully, we actually know what all of these do, so we know that it isn't an unknown mechanism. There is certain drugs you'll find in medicine where it's like, oh, this works, we don't know why. Kind of terrifying when you think about it. But you might be thinking, hold on, hang on, I don't know what the mechanisms of action of all of these are, and that's absolutely fine. You don't actually need to know the mechanisms of action to get this question right. So if we look at the vignette, it says here that the patient is suffering from hyponatremia. And this alone gives us enough evidence to figure out what the answer is. So hyponatremia is literally defined as a lower than normal level of sodium in the bloodstream. So from the vignette alone, we know that the only answer that makes sense is number 4, blocks sodium channels in a use-dependent fashion. So literally, you don't even need to know any mechanisms of action. Just think hyponatremia or hypernatremia, as the case may be. That has something to do with sodium channels. But we will go through this a little bit more just to help with that. So let's get this diagram over. Okay, so these are the sodium channels. And as you can see here we mentioned before, carbamazepine, valproate, these will bind to these sodium channels and they block sodium from going through them. So in the context of this particular question, just because I have had a little look at the mark scheme, it is referring specifically to the drug carbamazepine. Now, this drug is well known to cause hyponatremia. So carbamazepine, as we can see here, acts on the voltage-dependent sodium channels, which carry inward membrane current. So by doing this, this drug reduces the likelihood of developing an action potential. And when it's saying here use-dependent fashion, this is referring to the pharmacological property where the binding or action of a drug depends on the frequency or duration of its use. So in the context of a sodium channel blocker like carbamazepine, it means that the drug preferentially binds to sodium channels that are in the open or activated state. And this binding increases with repetitive or sustained neuronal activity. So all of that is just a fancy way of saying if the channel's open, it's more likely to bind to it. So we'll just go over the other ones. You have effusiximide. Man, I can say it fine. As soon as I'm recording, other people can hear me. Effusiximide, valproate, and gabapentin. These all do block calcium channels. In addition, valproate has the effect of actually enhancing GABA-mediated synaptic inhibition. So because it enhances this, we also know that it doesn't block it. It enhances it. But in relation to this, the fact that it does this, the fact that it activates on calcium channels, or the fact that it has an effect on GABA mediated, all of that has nothing to do with sodium. It wouldn't cause hyponatremia. So although, yes, these are mechanisms of actions that these drugs work on, they're not going to be causing hyponatremia. So you can rule those out. Okay? Hopefully that makes sense. I'm still learning to explain things. Like I said, if I've botched anything, this is my first one. Just leave a comment below, and I'll try and get back to you. Okay. In fact, I think probably just to visualize this, let's do this on a second page, shall we? Okay. Right. Penicillin 5. Penicillin 5 is part of the widely used beta-lactam class of antibacterial drugs. What is the mechanism of action of these drugs? So, does it inhibit the decarboxylation reaction involved in peptidoglycan utilization in the cell wall? Does it bind irreversibly to D-alanine, inhibiting the transglycylation reaction involved in cell synthesis? Does it inhibit a transpeptidation reaction involved in peptidoglycan utilization in the cell wall? Does it inhibit the transport of peptidoglycan to the cell wall? Or does it inhibit a transglycylation reaction involved in peptidoglycan utilization in the cell wall? Okay. So, beta-lactam antibiotics, which is what we're looking at here, include both penicillins and cephalosporins. So what I'm going to do, I'm going to grab another. In fact, I've stolen this one from Osmosis, which is another great resource if you're trying to learn. Okay, so beta-lactam refers specifically to this four-membered lactam ring structure that contains the beta-lactam moiety. So the beta-lactam ring is characterized by a beta carbon atom linked to both a nitrogen, so this one here, which forms the lactam ring, and a carbonyl group. So that's this C here, double bonded to the oxygen. So the whole reason this is important, so this arrangement is what confers the antibacterial activity of beta-lactam antibiotics, because these serve as the reactive site for binding to and inhibiting enzymes called penicillin binding proteins, or PBPs, and these are what are involved in cell wall synthesis. So bacterial cell walls are composed of peptidoglycan, which is this complex structure. In fact, hold on, let me probably make more sense if I get this image across as well. That makes more sense, doesn't it? Okay, so we have this structure. And these are made up of these sugar chains cross-linked by these short peptides. So during cell wall synthesis, precursor molecules are assembled into these long chains by enzymes called glycotransferases. These chains then cross-link to form a strong, rigid structure by enzymes called transpeptidases, and these transpeptidases are also what are referred to as penicillin binding proteins. And let's get a slightly more simplified image just to help understand this a little bit better. Here we go. So these enzymes are what are important. So beta-lactam antibiotics contain the beta-lactam ring in the structure, and these work by irreversibly binding to the active site of these transpeptidases, and they mimic the structure of the normal substrate, which is the D-ala end of the peptidoglycan precursor. Okay, so this binding inhibits the transpeptidase enzyme from forming cross-links between adjacent peptidoglycan chains. So this is what stops bacteria cell walls from binding together. So with transpeptidation inhibited, the bacterial cell wall loses its ability to perform these proper cross-links between the peptidoglycan chains. So these cross-links we can see here. As a result, the bacterial cell wall becomes weakened and unable to withstand the internal osmotic pressure, which leads to cell lysis and then cell death. So this is something that we would refer to as a bacteriostatic rather than bactericidal. It prevents the formation of new bacteria. It doesn't kill them specifically, but it prevents them from growing and proliferating. So it's a bacteriostatic type of antibiotic. So if we go back up to our questions, so the correct answer is a transpeptidation reaction. So the correct answer is number 3. So it's not number 1, as penicillin inhibits a transpeptidation reaction rather than decarboxylation. So we can rule number 1 out. It's not number 2, as penicillin mimics the terminal D-ala part of the side chain, which is recognized by the transpeptidization. It doesn't inhibit it, it mimics it. Okay, so it's not 4, because penicillin ultimately leads to fewer crosslinks, which reduces the strength of the cell wall and eventually causes cell lysis. So it doesn't inhibit the transport, so we can rule that one out. And then it's not number 5, as it inhibits transpeptidization rather than transglycosylation. So again, when you're thinking of penicillins, think beta-lactams. In fact, when you think of beta-lactams, think of penicillins and cephalosporins, and also think of transpeptidation. So think specifically, it is inhibiting this enzyme right here, and that's what stops the cell wall from forming. And as you're probably aware, penicillin's one of the first antibiotics. It's quite commonly used, a little bit less now, but it's a very commonly used antibiotic. So it's very important that you're aware of this as healthcare professionals. Okay, so question 3. Which of the following neurons utilizes a catecholamine? Jesus, I really can't speak today. Catecholamine neurotransmitter. So is that the preganglionic somatic neuron, the preganglionic parasympathetic neuron, the postganglionic parasympathetic neuron, preganglionic sympathetic neurons, or postganglionic sympathetic neurons? Okay, so let's start with basics. So what are catecholamines? So catecholamines are neurotransmitters which are derived from the amino acid tyrosine. So these include noradrenaline, or norepinephrine if you're American, adrenaline, or again epinephrine if you're American, and also includes dopamine. So straight away you should be thinking, okay, what system does adrenaline work on? So to help us understand this, I'm going to take an image from one of the other videos that I've created, which was made by my good friend, Doc Odsocks, who may be making some videos soon, actually. She's been helping me quite a bit. So we have this. So we have the peripheral nervous system, okay? So let's break this down. So the peripheral nervous system can be divided into the somatic nervous system and the autonomic nervous system. So the somatic nervous system refers to voluntary movements of skeletal muscles. So, think about, and that's why we made this little diagram. So think about kicking a ball, maybe giving peace signals or flipping someone off if you've really pissed off with them. And it also includes reflex arcs, so such as pulling your hand away quickly when you're touching something hot, or that knee-jerk reflex when the doctor hits your knee with a hammer. So the somatic nervous system makes use of nicotinic acetylcholine receptors, and as such this system primarily uses the neurotransmitter acetylcholine. So that is not a catecholamine, so you're not thinking adrenaline. So we can go up here and we can say it is not this. In fact, I'll leave that for a second, actually, because there's a little bit more I want to talk about. So the autonomic nervous system is regulated by involuntary bodily functions. So this can be divided further into the sympathetic and parasympathetic systems. So the sympathetic nervous system is usually referred to as the fight or flight response. I'm sure everyone's hopefully heard of that. And the parasympathetic system can be referred to as the rest and digest response. So it's quite a common colloquialism that adrenaline results in a fight or flight response. And you've heard it, boxers when they're going to go and fight, oh, we had the adrenaline ready to fight kind of thing. So this is partially true, but it doesn't tell us the whole story. And again, we're going to have to narrow our answer down a little bit more. So catecholamine includes adrenaline. Adrenaline is involved in fight or flight, and fight or flight is the sympathetic system. So again, we can rule out 1, 2, and 3, since they're referring to somatic and parasympathetic. So we know that it is a sympathetic system. But then is it preganglionic or postganglionic? And some of you might be asking me, what the hell does that even mean? Which is what I first thought when I first saw this type of thing. So again, we're going to get another diagram, which will hopefully help us to better understand this. Here we are. Oh, god damn it, it's a bit. Let me just make that a little bit shorter so you can actually see the whole thing. Okay, fantastic. Okay, so pre- and postganglionic refers to the location of the neurons in question. So preganglionic, so these are these two up here on both the sympathetic and parasympathetic side. So these two, preganglionic neurons are located in the central nervous system. So that's either in the spinal cord or in a specific brain region, depending on the division of the autonomic nervous system that you're referring to. So postganglionic neurons, so these ones, are located in autonomic ganglia, which are collections of nerve cell bodies located outside the central nervous system. In fact, hold on. Let's get another diagram to help understand this a little bit better. Like I said, diagrams really help me learn, so hopefully they can help you too. Okay, so this is kind of what I mean by the fact that adrenaline affects you, it being partially true. So you have the preganglionic neuron, so that's these ones that are from like the brain or the central nervous system, and then the postganglionics, it's gone to this autonomic ganglion, which then goes to the target tissue, whatever organ that may be. So the preganglionic neuron in both the sympathetic and parasympathetic systems both use acetylcholine, and that's what this part is mentioning here. So the preganglionic, these ones are both releasing acetylcholine. So the difference comes in the postganglionic neurons. So the sympathetic system here, as you can see it here, the fight or flight response, it's releasing norepinephrine or noradrenaline, whereas the postganglionic on the parasympathetic side makes use of either acetylcholine or nitric oxide. So the way to kind of remember, parasympathetic uses acetylcholine both before and after, whereas the sympathetic uses acetylcholine before and adrenaline, or norepinephrine rather, noradrenaline afterwards. Okay, so if we go back to the original question, so it's not 1, as the as the somatic nervous system does not have preganglionic and postganglionic divisions at all. So the ganglion referring to the group of nerve cell bodies located in the autonomic nervous system and sensory system. So that's not something part of the somatic system at all. It's not 2 or 3, as both pre- and postganglionic parasympathetic neurons utilize acetylcholine as a neurotransmitter. And, well, acetylcholine is not a catecholamine, so we know it can't be that. And then the same goes for number 4. So the preganglionic sympathetic neurons also make use of acetylcholine, not a catecholamine. So the only answer we have is postganglionic sympathetic neurons. So yeah, these ones here, these are the ones that use norepinephrine or noradrenaline for the fight or flight response. And that gives us our answer. Okay. And my mic decided to stop working, so let's start this again. Love technology. Right. Question 4, again for me. Okay, so a histopathologist is sent some tissue samples to examine, including a sample of blood. Blood belongs to which primary tissue type? So is that plasma tissue, nervous tissue, connective tissue, blood tissue, or epithelial tissue? So this is a little bit of a trick question, and you might be thinking, hang on, it's not obviously just going to be blood tissue, right? And you're correct, it is not blood tissue. So there are 4 primary tissue types, and let's bring over a little diagram to help you better visualize this. So the 4 primary tissue types we have are epithelial tissue, nervous tissue, muscle tissue, and connective tissue. So blood tissue is not a type. Blood tissue is not something. And also plasma tissue is not a tissue type. So straight away, we can rule those ones out. So now we want to figure out, okay, so what is blood? Which of these 4 is it? And for that, I'm going to get another diagram because I'm a very visual learner. Okay, so let's go through these, shall we? So nervous tissue. Nervous tissue is composed of neurons, so nerve cells, and neuroglia, so those are the supporting cells. These form the brain, the spinal cord, nerves, and these are what are responsible for transmitting electrical impulses and coordinating sensory input. Again, that includes like integration, motor output, that kind of thing. So nervous tissue doesn't include blood. Epithelial tissue, this stuff here, covers body surfaces. So it lines body cavities, hollow organs, it forms glands, and the functions it serves are for protection. So again, think, skin, use that for protection. Absorption, so the gastrointestinal tract, absorption, secretion, and sensation. So again, tactile sensation on your skin. This doesn't include blood. I don't know how else to word that one. Yeah, it's kind of like what lines certain organs. So again, yeah, skin, GI tract, that kind of thing. Doesn't include blood. So the only answer we have left, because it didn't actually say muscle tissue as one of the answers, is connective tissue. So connective tissue provides support, structure, and connection, as the name implies, to other tissues and organs. And this actually encompasses quite a wide range of different tissue types. So you have connective tissue, dense connective tissue, adipose tissue, so kind of fat and stuff, cartilage, bone, and blood. So I know that seems a little bit strange, but blood is actually considered a type of connective tissue. So the correct answer is number 3, connective tissue. Let's just make this nice and green and nice and bold to stand out, and then we'll cross out the other 2. Need to get faster at doing that. Just for the sake of completion, I'm certain you probably know what this is, but we'll just cover it. Muscle tissue is responsible for generating force and producing movement. So there's three types of muscles. So you have the skeletal muscle, which is the one that attaches to bones, and that's what facilitates voluntary movements. Smooth muscle is found in the walls of hollow organs and blood vessels, and those are involved in involuntary movements. And then finally you have cardiac muscle, which makes up the heart, and, well, I'm certain you know, heart is responsible for pumping blood, but is not blood itself, which seems obvious. But yeah, cardiac muscle, heart, not blood itself. Not blood itself. Not blood itself. And another mic cut, but this actually gave me a big brain idea. This probably makes more sense to orientate this landscape, doesn't it? I should have started with that. Right, question 5. Which of the following statements correctly explains the difference in solubility in water between fibrous and globular proteins? So globular proteins are more soluble because their hydrophilic amino acids are orientated outwards and their hydrophobic amino acids inwards. Globular proteins are less soluble because their hydrophobic amino acids are orientated inwards and their hydrophilic amino acids outwards. Globular proteins are more soluble because they do not contain any hydrophobic amino acids. Fibrous proteins are more soluble due to their spherical shape, and fibrous proteins are less soluble due to their spherical shape. So let's just start by visualizing this. Okay. Yes, I am just stealing images from Google Images. Okay, fibrous proteins are these green ones on the left-hand side. So these are made up of polypeptide chains that lie in parallel to each other and form a fiber, hence fibrous. In fact, probably a better way of visualizing this. So this is like on the single subunit level, but if you see them all bunched up together, so yeah, they form these long chains like this, whereas globular proteins, you have a bunch of these and then they tend to like kind of join together when you get to the tertiary and quaternary structure. So globular proteins do also have these polypeptide chains, but these intertwine with each other to form a more kind of spherical shape. And then there's another, this is from actually a chemistry lecture, I believe. And this is quite, heavy biochemistry, to be fair. So fibrous proteins, I mean, you can kind of see from here, their purpose is to form some kind of structure. So some examples of this, so think like keratin, which can be found in hair. It can also be found in the outer layer of skin. And collagen as well, that can be found in tissues. It can be found in skin tissues as well. So just a few different types of fibrous proteins to think about. So you've got collagen, myosin, fibrin, actin, keratin, and elastin. So these help to form certain structures, and something that can hopefully help you remember. These tend to form things like bones, tendons. They form the walls of blood vessels. Silk, apparently. That's very odd. Not really related to humans, but yes, silk is a fibrous protein. Whereas globular proteins, these include things like enzymes, insulin, immunoglobulins, and hemoglobin. So these proteins' function is to play more of a role in metabolic reactions. And, there is some variation, but generally speaking, and what you need to remember, fibrous proteins are insoluble in water, whereas globular proteins are soluble in water. And then if you just think of some of the structures that you can make. So fibrous protein makes bone. Bone is obviously going to be insoluble. Globular proteins make blood. If you drop blood in water, that's going to be soluble. So that's kind of the way I remember it. So with that, we can answer, or at least rule out 2 questions. So fibrous proteins are less soluble than globular proteins. However, fibrous proteins are not spherical shaped. So we can cross. Where's the bloody cross out gone? There you are. So we can cross this one out. Now, fibrous proteins are more soluble due to their spherical shape. Again, fibrous proteins are not spherical, and they're also less soluble, so we can rule this one out. Globular proteins are more soluble because they do not contain any hydrophobic amino acids. So yes, globular proteins are more soluble, but it's not because they lack hydrophobic amino acids. So we'll scroll down again. So let's get a couple more images. So when you have this single unfolded polypeptide chain, you have both polar and unpolar amino acids. And these kind of like form, like when you have the chain together, they contain both hydrophilic and hydrophobic amino acids. However, when these unfolded polypeptides start to form into the tertiary and quaternary structures, their conformation in an aqueous solution, so in water, it will have, in fact, just to get this up, so you'll have the hydrophobic region, so that's the region that doesn't like water, that will form inwards, and then the polar side chains are on the outside, those are the ones that like water, and they can form bonds with water. So they have, it would be an incorrect statement to say they don't contain hydrophobic amino acids. They do, it's just the way that they're formed changes that. So we can rule this one out. And then globular proteins are less soluble because their hydrophobic amino acids are orientated inwards and the hydrophilic amino acids are orientated outwards. And then we have globular proteins are more soluble because the hydrophilic amino acids are orientated outwards and the hydrophobic amino acids orientated inwards. So the correct answer would be number 1. So you have the sorry, I'm getting so tongue-tied. Not used to doing this all at once. So globular proteins are spherical in shape, and they are soluble in water due to their hydrophilic amino acids being orientated outwards, while their hydrophobic amino acids are orientated inwards. So again, if we go down to this diagram, so the part that doesn't like water is on the inside, and the part that likes water is on the outside. And that's why they're soluble in water, because they can mix. They can mix together like that. Sorry, I know the answer, but then I was just double reading through this and my brain just went, ugh. Right. Okay. Question 6. Okay, so which of the following is the amino acid that is found at every third residue in collagen? This one annoys me a little bit, because this is just one that you kind of, you know or you don't. It's normally something that you can work logically through. But I suppose it gives me a good excuse to talk a little bit about the amino acids, which, again, I'm going to assume, because this is the first question, that we're going to build our knowledge up. I probably won't be talking about this as much in depth of the later ones, but I'll just basically start from scratch. So you have 20 amino acids, and these are the 20 amino acids you need to know about. And these amino acids are divided into different groups, whether or not they're polar, non-polar, positively charged, negatively charged, that kind of thing. And so if we look at this, one thing you might notice, so hydroxylysine, hydroxyproline, hang on, that seems weird. So proline and lysine are both amino acid. However, hydroxyproline and hydroxylysine aren't. So hydroxylysine and hydroxyproline are formed due to what is known as a post-translational modification. This refers to chemical modifications that occur on a protein after they have been synthesized and are folded into their native conformations. So in this case, the protein in question has undergone what's known as a hydroxylation reaction. In fact, do I have a diagram for this? No, I don't. I thought I did. My apologies. So they undergo what's known as a hydroxylation reaction. And this is basically when you have an OH group that is added to the side chain of the protein. And post-translational modifications, that plays a crucial role in kind of like regulating protein structure and functioning, plays a crucial role in cellular signaling and physiological processes. But all of that is very much beyond what we need to know from now. All we need to know is that these are post-translational, so these are not amino acids. So why have I made that green? So, make it black again. So it can't be either of these because these are not amino acids. And then there's also this one. Hang on, this doesn't look like an amino acid either. So tropocollagen is a subunit of collagen. It's not an amino acid, but a protein. So we can also rule this one out. And this leaves us with proline and glycine. And unfortunately, at this point, it's just kind of a matter of knowledge. If we scroll down here, so both proline, which is here, and glycine, which is here, these are both non-polar amino acids. So we can't deduce from this which one it would be. So the correct answer to this is actually glycine, and I'll kind of talk you through this because this is something that you'll probably need to remember. And it's again just one. It's just the memory one. There isn't any logical deduction that can really be done. So, when we're forming collagen, it makes this fibrous protein, right? And this structural framework is built up of certain repeating patterns. So the primary structure is categorized by what's known as a repeating tripeptide sequence, where glycine is found at every third position. So if you were to look at this from more of a bioinformatic kind of way, so you can see here, every third position is glycine, and that's what this refers to. So it's not always proline, as we can see. Say we have. Yeah, proline are the yellow parts, but it's not always that. So this is what's known as a glycine XY repeat. X usually denotes a proline amino acid, but not always. And this repeating pattern is essential to the formation of the collagen's triple helical structure, and that's what helps contribute to its strength and its stability. So glycine, because it's in every third one, makes up about 33% of collagen. Some of the proline is modified to hydroxyproline. So proline makes up between 13 to 17% of collagen, depending on what source you're using, whilst hydroxyproline makes up about 11%. So proline is the second most common amino acid, whilst glycine is the most common. And again, that's just one you have to remember. There's no other way around it, unfortunately. And it's a bit of a weird to go that much into biochemistry. It depends on how mean the examiners are feeling. Thinking of how mean the examiners are, this question annoys me because I actually know what the correct answer is. I have the mark scheme. And it's I'm not sure I fully agree with it. And I'll kind of explain my reasoning as we go through it. But don't worry about this. It's still a good learning opportunity. Okay, so let's just separate this out. So a 24... Sorry, let me just move that up. A 24-year-old woman presents the local pharmacy asking for the emergency contraceptive pill. She had unprotected sexual intercourse 5 days ago and says that she may be pregnant. What term would be used to describe the stage of development of the embryo in this case? So is it a blastocyst, a morula, an ovum, a trophoblast, or a zygote? So let's kind of work through this logically and go through all the stages. So after the bedroom dance is complete, sperm is able to reach the fallopian tube where fertilization usually occurs. And how long this take can vary. It's usually within a few minutes to a few hours after ejaculation. So For the purpose of understanding this, let's just assume it happens immediately, just so we can understand it a little bit better. Okay, so this is kind of the embryological development as we go through. So if we assume that fertilization happens at day 0, as is denoted in this diagram, the egg at day 0 is known as an ovum. So we can rule out ovum. From day 1 to day 3, the collection of cells forms what's known as a zygote. So we can rule that one out. At day 4, it is referred to as a morula. And this is the 16 to 32 cell stage, which occurs around day 4. And so, yeah, this is the morula. By day 5, the morula now is formed into what is known as a blastocyst. And the trophoblast, which was one of the answers, refers specifically to this specialized layer of cells that form the outer layer of the blastocyst. This layer is needed for the implantation stage of the embryo into the uterine wall, which subsequently forms the placenta. So, I would personally say that the correct answer is a blastocyst. However, I have access to the mark scheme, and I know that the supposed correct answer is actually morula. And this is because it assumes that fertilization takes place exactly 24 hours after ejaculation has occurred. So we are supposed to assume we take 1 day off, that the embryo is at day 4 of embryo development. So this question annoys me because if you look at the actual literature, and it depends on what source you quote, but the time it takes from ejaculation for fertilization to occur varies because humans are not an exact science. There is variation. And this time can be anything from a few minutes to 30 hours. So this question is getting us to make the assumption that the process took exactly 24 hours, which is, it's arbitrary. It should have been worded very differently and just say, what is the embryo at stage 5? It shouldn't have done this whole spiel about they had sex 5 days ago, where is it now? It really bugs me because if you look at the literature, a lot of times when it's like saying when the morula is formed, it will say between day 4 to day 6 because there's variation. So it's a very unfair question in my opinion, and it expects you to think exactly the way the examiner is. Yeah, sorry, I know I'm just bitching. Hopefully, regardless, if you come across a question like this in your exams, it won't be worded this way, it'll be worded a little bit better, and hopefully you can still use this little diagram here to kind of understand it. So think ovum, zygote, morula, blastocyst, and that will hopefully help you with embryology if they don't ask a silly question like this. So yes, allegedly the correct answer to this is morula, although personally if you put blastocyst, I put blastocyst, is all I'm saying. And again, if I'm being way off the mark here with this, please let me know, but I don't like the way this question is worded. And unfortunately, that is just how some medical questions are worded, and that's why they have the mark scheme so low, to account for shitty questions, to be honest. Anyways, you didn't come here to hear me bitching, you came here to learn some medicine. Okay, so let's move swiftly onwards to question 8. Okay, which of the following properties of cardiac muscle enables the propagation of action potentials? So is it the spontaneous electrical activity that only starts from the primary pacemaker, the sinoatrial node, the sinoatrial node acting as an ectopic pacemaker, the ventricular action potential acting as the sole action potential in the heart? Is it that the cardiac muscle is arranged in a syncytium, or individual cardiac myocytes lacking spontaneous electrical activity? Okay, so let's get a picture of the heart up, shall we? Okay, so this is a good place to start. Because I know this is a question that several of my friends struggle on. So, You have electrical signals start at the sinoatrial node, and this is located in the right atrium near the junction with the superior vena cava. The SA node serves as the heart's natural pacemaker, so this generates rhythmic electrical impulses, and that's what initiates each heartbeat. So the electrical impulses generated by the SA node spread through the atria, which causes them to contract and push blood into the ventricles. And then you have the atrioventricular node here. So this is located at the lower part of the intral septum. And the AV node acts as a relay station for electrical impulses. It delays the transmission of the impulses slightly to allow for the atria to finish contracting before then the ventricles contract. After this, it passes through to the bundle of His. So these electrical impulses travel down the bundle of His, and it's called that because it's basically a bundle of specialized fibers, and these extend from the AV node into the intraventricular septum. And you also have, you have the left and right bundle branches. So the bundle of His divides into the right and left bundle branches, which then extend into each of the respective ventricles. And then once you get a little bit further down here, the bundle divides further into what are known as these Purkinje fibers. So these spread throughout the ventricles, and then these fibers rapidly conduct the electrical impulses to the myocardium of the ventricles, and this is what helps cause coordinated contractions and ejections of blood from the heart. So to understand this, we kind of need to understand the way that these electrical impulses are formed. So with this diagram, let's go through this. So in your heart, you have what are known as these intercalated discs. These are specialized junctions found between adjacent cardiac muscle cells or cardiomyocytes, and these discs are what help coordinate the contraction of cardiac muscles and transmit electrical impulses between cells. So these consist of several components. So you have your gap junctions. So these are channels that allow for direct electrical communication between neighboring cardiomyocytes. So these are what permit the rapid spread of these action potentials from one cell to the next, and this helps ensure synchronous contraction of the entire myocardium. So these gap junctions here. In addition to that, you have these desmosomes. So desmosomes are anchoring junctions that provide mechanical strength and stability to these intercalated discs. So they anchor adjacent cells together, and that helps withstand the mechanical stress generated during the cardiac contraction. So as you can imagine, the heart is always going, so it's under a lot of stress. Okay. So that's quite a whole lot of information I've thrown at you. There's a few little more bits. So you have what's known as the fascia adherens. So it's not actually on this diagram. That's annoying. So the fascia adherens are another type of anchoring junction. So these are found within intercalated discs, and they anchor the actin filaments of the cardiac muscle cells to the plasma membrane, and that helps to transmit contractile forces between cells. So these components of intercalated discs facilitate electrical coupling and mechanical cohesion between adjacent cardiomyocytes, and this is what allows for the efficient propagation of action potentials and synchronized contraction of the heart cells. And the way that this is all kind of described is in a syncytium. So in the context of cardiac muscles, this refers to the function formed by the interconnected cardiac muscle cells. So cardiac muscle cells are electrically coupled through these gap junctions, and that allows them to behave as a single functional unit, and that's really important. They're a single functional unit. When one cardiomyocyte is stimulated to contract, the action potential rapidly spreads through the gap junction to neighboring cells. And this is what results in the coordinated contraction of the entire myocardium. You can't see me, I'm doing a lot of hand movements here, trying to visualize it. So these synchronized contractions ensure efficient pumping of blood by the heart, and that is facilitated by these intercalated discs that connect the adjacent cells together. So in this way, the cardiac muscle is often described as a functional syncytium due to its coordinated electrical and mechanical activity. So that's a whole lot of information. So let's go back up to this and kind of like run through it and just go through it one at a time, basically. So spontaneous electrical activity that only starts from the primary pacemaker, the sinoatrial nerve. So this is incorrect because spontaneous electrical activity can arise from the AV node or from tertiary fibers. So think of the bundle of His and the Purkinje fibers. So if the normal electrical conducting pattern is compromised, you have the bundle of His and the Purkinje fibers can help propagate it forward. So this would be an incorrect statement because it doesn't only start from the primary pacemaker. So we can cross that one out. Okay, so 2 is incorrect because the SA node is the primary pacemaker. When it's talking about it being an ectopic pacemaker, yeah, and any group of pacemaker cells that can generate spontaneous electrical activity abnormally. So again, we're thinking tertiary fibers. Those are known as ectopic pacemakers, but the SA node is the primary pacemaker. So again, it's kind of, if you want to kind of like look for like the wording. So the fact that it's ectopic is what rules that one out. Okay, the ventricular action potential as the sole action potential of the heart. This is also incorrect because there are 2 types of action potentials in the heart: the sinoatrial and the ventricular action potentials. And number 5 is also incorrect because even when isolated, cardiomyocytes have the ability to maintain that contractile functionality. So we can rule out number 5 and we can rule out number 3. So again, because all of these kind of sound like they're mostly correct, but you have to be quite careful about like the individual wording of it. So the correct answer is cardiac muscles arranged in this conformation. Okay. Okay, so question 9. A 37-year-old female presents to her GP as she has been coughing up green sputum for the past 10 weeks. She has lost around 4 kilograms in weight over the past month without intending to do so and has been feeling generally unwell. A sputum sample is sent to the lab for analysis and the report reads acid-fast bacilli detected. The doctor concludes that she has Mycobacterium tuberculosis infection and treats her accordingly. She recovers well. 6 months later, she returns to the doctor as she is coughing again. The woman is concerned that her tuberculosis infection has returned, but the doctor reassures her that it is a simple viral infection. Which of the following is a manifestation of secondary tuberculosis? Okay. So to answer this one, we kind of need to know what all of these diseases are, as well as what is meant by acid-fast bacteria. So let's start with that first. So acid fastness is a physical property that gives a bacteria the ability to resist decolorization by acids during the staining process. So this is when we're looking at gram staining. So this is kind of like the classic example, isn't it? You've got gram negative and gram negative being pink, gram positive being purple. So when you think of something that can resist this, there's a few different ones you can be thinking about, but the one you need to absolutely be aware of is obviously Mycobacterium tuberculosis. That's what it's mentioned. So the reason that Mycobacterium tuberculosis resist this is because it has this extra waxy lipid layer on its outer surface. So normally when we visualize tuberculosis, it's with a different staining technique known as a Ziehl-Neelsen stain. So if ever you hear Ziehl-Neelsen, start to think this is probably tuberculosis. And so this is just, I mean, this is a little bit beyond what we need for this question, but again, it's just, I want to help explain everything around it just so we can fully understand what's going on. So you have a slightly different technique, which I don't think you need to really understand the technique too much unless you're a microbiologist. Obviously, it's very important for them. But it's more just being aware that if you hear this, think tuberculosis when it comes up in a question. So now that we understand that, let's go through all the different diseases. So let's start from the top. So Pick's disease. So Pick's disease is what's known as frontotemporal dementia. So I think a good way to kind of look at this, and yes, we're going well beyond this one. But I find the best way to know with these questions what's going on is to understand. There's nothing worse than like, I think it's this question, but I don't even know what the other things are. So I just kind of want to, go through all of these are. Okay, so it's named frontotemporal dementia due to the presence of abnormal protein aggregates known as Pick bodies or Pick cells. So if I get... So yeah, you get these Pick bodies if you were to look at it histopathologically. So this image shows Pick bodies in two separate areas. So slide A and B shows them in the dentate gyrus, and slide B shows them into the enteral cortex. So these are different sections of the brain. So these aggregates are composed of an abnormal form of a protein called tau, which is a protein you will hear quite a lot about whenever you're discussing neurodegenerative disorders. And specifically, so with Pick's disease, so normally if you're looking at like a brain CT scan, you'll see it like this. When you're looking at frontotemporal dementia, you will see this. And so if we look at this, just see what we're looking at, especially around the frontal temporal lobe, you can see there is a lot of atrophy. There is a lot of degeneration there. Like the brain has literally atrophied down, especially the white matter region. So it's not a fun disease. It's really quite nasty. But that said, so Pick's disease is associated more with older individuals. It tends to first manifest as behavioral and personality changes around midlife, and then slowly progresses worse as an individual gets older. This, to the best of our knowledge, has nothing to do with tuberculosis, so we can rule question 1 out. The next question we have is Parkinson's disease. So this is a neurodegenerative disorder, but rather than it being associated with tau proteins, it's more associated with the gradual loss of dopamine-producing neurons within the substantia nigra of the brain. So quite a famous sufferer of this is actually Michael J. Fox, the kid from Back to the Future. Obviously, well, he's not a kid anymore; he's a man. And he set up the Michael J. Fox Foundation to fund research. And I just wanted to mention that because I think it's amazing and something I'm incredibly interested in and is wonderful. It's not obviously wonderful that people suffer from Parkinson's, but it's wonderful that there's more awareness being raised about it and more research going into it. So Parkinson's is characterized by a triad of bradykinesia, and that refers to the slowness of movement, rigidity, resting tremor, and it leads to the formation of these things. Come on, Google. So it leads to the formation of Lewy bodies. And unfortunately, you might have actually heard of this, even if you're not medically related, because Lewy body dementia was what Robin Williams also suffered with. So it's horrible. There's no nice way of putting it. So Lewy bodies are these. So Parkinson's tongue twisted. Parkinson's disease leads to the formation of these Lewy bodies, which are composed of a protein called alpha-synuclein. This forms insoluble fibrils that accumulate inside the neurons. So this protein is normally found in the presynaptic terminals of neurons, where it plays a role in regulating neurotransmitter release. In Lewy bodies, alpha-synuclein undergoes abnormal folding and aggregation, forming insoluble fibrils that accumulate within neurons. So these ones here. Again, more associated with individuals that get a bit older, and as far as we're aware, has nothing to do with tuberculosis, so it's not Parkinson's disease. The next one we have is plumber's disease. And I really wish it'd be so much easier if they just named it something more conventional. But again, it's important that you know this, because certain medical conditions, although we're getting a little bit more medical with it, like the eustachian tube in the ear, for instance, you now tend to refer to as the pharyngotympanic tube. It's important to remember that not everyone's going to use that terminology, and you're going to be working with people that maybe use different terminology. So that's why we have to learn all this weird stuff. So, plumber's disease is also known as toxic nodular goiter or toxic multinodular goiter. And this is a thyroid disorder characterized by the presence of one or more nodules or lumps in the thyroid gland. These nodules are overactive and produce excessive amounts of thyroid hormones, leading to hyperthyroidism. This is a purely endocrine disease, so again, has nothing to do with tuberculosis. And then we come on to Paget's disease. So Paget's disease is also known as osteitis deformans, and is a chronic bone disorder characterized by abnormal and excessive remodeling of bone tissues. So in Paget's disease, bone remodeling occurs at an accelerated rate, leading to the formation of new bone that is structurally abnormal, enlarged, and weaker than normal bone. So it causes this kind of bent legs, this kind of like warped legs we can see here. So Paget's disease is not fully understood, but we do believe it to be a combination of genetic and environmental factors. Again, nothing to do, as far as we're aware, with tuberculosis. So that leaves us with one answer left. Pott's disease. Pott's disease is quite literally known as spinal tuberculosis. It is the most dangerous form of musculoskeletal tuberculosis because it can cause bone destruction, deformity, and paraplegia. Pott's disease usually involves the thoracic and lumbosacral spine, but yeah, it's basically when you get tuberculosis that goes into the spine and it causes this degeneration. So again, the hardest part of a question like this is just remembering all of the weird names they have for everything. But hopefully, you learned a little bit there about Ziel-Neeson and whatnot, because that always comes up in exams. You're almost always going to hear something about Ziel-Neeson, and hopefully now you think, oh, tuberculosis. So yeah, all of these wacky and wonderful names, we can rule all of these out, and we have Pott's disease as our answer. Okay, so last one. When I get there. Okay. What is the first line antiemetic used to prevent nausea and vomiting in a patient receiving chemotherapy? Is it ondansetron, cyclizine, Zonve, domperidone, or metoclopramide? So again, this is a question you kind of just need to know, unfortunately, because all of these are used to treat nausea and vomiting. So I'm just going to give you the answer. Ondansetron is known as a 5-HT3 receptor antagonist. So 5-HT3 receptors bind to serotonin. And I think a good way to remember this, and again, a lot of medicine is kind of just, to be honest, just forming association between different things because there's just so much to remember. So the chemical name for serotonin is literally 5-hydroxyltryptamine. So that's something you can use to help remember it. So ondansetron works by blocking serotonin receptors in the gastrointestinal tract and the central nervous system. In fact, let's make this a little bit bigger so it's easier to visualize. So they block serotonin receptors in the gastrointestinal tract and the central nervous system, which therefore reduces the sensation of nausea and the urge to vomit. So ondansetron is usually taken 30 minutes before the start of chemotherapy to help prevent nausea and vomiting. It's usually, I say that, I mean, when you look up the guidelines, they will say that it's usually effective, especially when it's combined with another drug known as dexamethasone, which we'll be probably covering in episodes in the future. Okay, so just to help with the understanding of the other drugs, and hopefully, because although, okay, we have the answer for this, but it's important to know what these other ones do because they're probably going to come up. So intravenous cyclizine is usually the second line agent used to prevent and treat post-operative nausea and vomiting. So not first line, second line. And this is, a lot of medicine is, you kind of just like develop an association, like certain disorders, certain diseases, first line drug, you associate that with the disorder. And it's just something you kind of need to do. Works by blocking the action of histamine at H1 receptors in the vestibular system of the inner ear and the brain. So that's cyclizine. Then you have Zonvia, is a combination medication used for the treatment of nausea and vomiting in pregnancy. So it contains two active ingredients, doxalamine succinate and pyridoxine hydrochloride, or vitamin B6. So doxalamine is an antihistamine with antiemetic properties. Pyridoxine, also known as vitamin B6, is a water-soluble vitamin that plays a role in various metabolic processes in the body. So in the context of Zonve, the drug we're looking at, pyridoxine is included to supplement vitamin B6 levels, as deficiency of this vitamin has been associated with an increased risk of nausea and vomiting in pregnancy. So if we go up to our ones, so cyclizine, second line, Zonvia, pregnancy. That's the way to kind of associate it. And then, just because I find this quite helpful, okay, so the next one we have is domperidone. So this medication is primarily used to relieve symptoms of nausea and vomiting as the others. The way this one works is a little bit different. So this one is a dopamine receptor antagonist. So it targets dopamine D2 and D3 receptors. It blocks these receptors, which results in increased motility of the stomach and intestines, and that helps to decrease nausea and vomiting. It's usually used for conditions such as renal failure and Parkinson's disease, again, because it's a dopamine receptor antagonist, the way it interplays. And it is also what's usually used if the symptoms of your nausea and vomiting aren't known. You'll normally go to domperidone. And then finally we have metoclopramide. So this one is another dopamine receptor antagonist, but this is used more to treat delayed chemotherapy induced nausea and vomiting. So again, just if we get the 5, so ondansetron, first line, cyclizine, second line, Zonvier, pregnancy, domperidone, renal and Parkinson, and then metoclopramide is more when you have delayed. So first line, second line, pregnancy, renal, delayed. And that's just kind of the way I remember it. Anyways, this has been my first one. I know this was... Hopefully I'll get better with these and get a little bit less stumbling as we go on. But hopefully, regardless, you found this helpful. I'll just leave it here. Thank you very much for watching. Hopefully you learned something, and I'll see you in the next one.